Before we jump into the rest of Chapter 13, covering slides 10 through 23, I'd like to actually zoom to the very end of our presentation and look at this last slide. Okay, so for today, the material that we're going to cover, I don't expect you to know more detail than what we're going to cover in this presentation. What I've done down here is I've given you a table of all of the different classes of antibiotics that we are going to go over. That lists all of the ones that we're going to talk about, but some of them are bold and some of them are just regular type. So the ones that are in bold, penicillin, cephalosporin, and sulfonamides are going to be drug classes that you need to know a great deal about their mechanism of action. We're going to spend some additional time talking about these and really understand how it is they work to kill bacteria without inhibiting our own cellular processes. The other ones we're going to touch on just briefly. For those, I am going to tell you straight up that I will have that as that information as a matching question on your exam. So know a lot more information about penicillin, cephalosporins, and sulfonamides for additional multiple choice questions and things like that. Excuse me. But the other stuff we're going to cover just so we have an idea of some of the different types of antibiotics that we can have to fight off our pathogens. All right, so when we talk about all of these different antibiotics that we're going to be addressing today, the idea here is that these antibiotic drugs are going to inhibit microbial growth or flat out kill our microbes by disrupting some critical physiological function within the cell. We have to disrupt something that the bacteria absolutely needs in order to survive. And we have to do that in a selectively toxic manner. We have to make sure that our cells are not going to be kind of collateral damage. So when we start talking about these antibiotics, there's kind of some general physiological processes that we're going to target. So we'll see that we're going to talk about cell wall synthesis. Well, peptidoglycan is a great target for antibiotics. When we talk about our cell or our plasma membranes, remember we have to be really careful here because our phospholipid bilayers that make up our membranes are very similar to those that make up prokaryotic cell membranes. DNA synthesis, where we copy our DNA, RNA synthesis, where we read our DNA and make RNA, and then that protein synthesis by ribosomes, where RNA is made into a protein. And then additionally, we can look at a variety of different metabolic processes that we can disrupt. Most commonly, when we are going to be using an antibiotic that targets a metabolic process, it's really the enzymes that we're going to be targeting. So when we look at this particular enzyme here, and if we can disrupt the binding of the substrate and the catalyzation of that reaction, we can shut down critical metabolic pathways. So this is kind of an introduction into the different categories that we have of antibiotics. So we're going to start off with antibiotics that target the cell wall. This structure is ideal for selectively toxic antibiotics because the cell wall is composed of peptidoglycan. Mammalian cells, you are a mammal, so your cells never have PTG within them. And so if we can disrupt if we can disrupt either its assembly or the cell wall itself in any way, we can cause the cell to burst. So real quickly, I'd like to review how we make our cell walls. So in this diagram, we have the inside part of the cell here, the cytoplasm, and then here is the exterior portion where we're actually going to build our cell wall. Our subunits of our cell wall are composed of a NAM monosaccharide with a short peptide chain extending from it and a NAG monosaccharide. These two come together to form a disaccharide. That's all done within the cell. Well, we have to get those individual building blocks outside. So that individual subunit is going to pass across the plasma membrane, where we're going to have a number of enzymes that are going to go to work creating the covalent bonds that assemble that one single porous molecule. The enzyme that we talked about as our example was transpeptidase. We are going to see that it is one of many enzymes that are responsible for forming the cell wall. All right, so we're going to talk about the different types of antibiotics that inhibit cell wall synthesis. We said that peptidoglycan is an awesome target for these drugs. We don't have it, and our prokaryotic cells require that cell wall to prevent bursting. If we disrupt it somehow, then the cells will burst and they will die. So we've mentioned a couple of different antibiotics up till now. We're going to begin by talking about penicillin and cephalosporin. These are part of a group of drugs called beta-lactam drugs, and there are a number of different kinds of beta-lactam drugs. But penicillin, cephalosporin, and all of these drugs collectively have a similar structure within their compound. It's called a beta-lactam ring. That's why they have the name beta-lactam drugs. So here we see in our picture of penicillin, this is our beta-lactam ring. Notice that we have some additional functional groups off of this. There is an R group over here. This is essentially a position holder. We can change what's here to make different forms of penicillin. Cephalosporin is down here. It is a beta-lactam drug because it has that beta-lactam ring. Slightly different slightly different functional groups from that. And then we can see those R groups as our placeholders. So we can have different forms of cephalosporins as well. We've talked about how penicillin functions, its mechanism of action or its mode of action. We've said that transpeptidase, our enzyme that is responsible for catalyzing the formation of our peptide bridges, is going to have penicillin come in and bind to its active site. And it's going to sit in that active site, preventing transpeptidase from forming the covalent bonds between our peptide chains. We don't make our peptide bridges and the cells will burst. So we're going to kind of take a big step back and realize that we were looking at a very specific example. So all of these beta-lactam drugs have that beta-lactam ring, and that is what is sufficiently matched to that active site to go in and bind to the active site. We're going to give you a little more information about that process. It is called competitive inhibition. So what happens is that those beta-lactam drugs competitively inhibit they kind of outcompete the peptide chains for that active site. So when we talk about competitive inhibition, that's what it means. When we competitively inhibit an enzyme, we just sit there and we kind of occupy that space. And so as long as these beta-lactam drugs are sitting in the active site, that enzyme cannot catalyze the reaction. So transpeptidase is one of many protein enzymes that are responsible for forming parts of our peptidoglycan cell wall. So recall that all enzymes are forms of proteins. Okay, so an enzyme is just a special type of a protein. So when we start talking about lots of these different enzymes, well, these are ultimately proteins that penicillin and other beta-lactam drugs are going to target to competitively inhibit. So that broader group of enzymes that includes transpeptidase are considered to be PBPs. Those enzymes are called penicillin binding proteins. Okay, so transpeptidase is one example of a PBP, a penicillin binding proteins. Because these drugs are going to competitively inhibit the enzymes that form the covalent bonds, they don't have any effect once the cell wall has been made. So these drugs are only going to be effective against actively growing and cells that are undergoing binary fission actively, because it's those cells that are constantly needing to make new cell wall components that are going to be affected. So if we have cells that are fully formed with their cell wall, they're fully mature, they would not be affected by this particular drug. By this particular drug class. It only will be effective against actively dividing cells that are in the process of making their cell walls. All right, so when we start looking at these beta-lactam drugs, we are going to see that they are most effective against certain groups of bacteria. So recall that gram-positive bacteria will sometimes have just a cell membrane and a thick layer of peptidoglycan. Remember, these guys stain purple in our gram stain. So I've pulled this diagram over here from a different textbook. I like it because it's nice and simple. But this is what we have, a gram-positive cell. We've got our cell membrane, and then we've got our thick layer of peptidoglycan. Well, recall that this is a very different setup from our gram-negative cells, which stain pink following a gram stain. They have two membranes, that outer membrane and that inner membrane. The space between the two, the periplasmic space, is where we find that thin layer of peptidoglycan. Well, the reality is that that outer membrane of our gram-negative cells acts to really block access of our beta-lactam drugs from getting to the enzymes that are building that cell wall within there. So these particular drugs are generally not very effective against our gram-negative cells because of that outer membrane. But additionally, the penicillin binding proteins, the PBPs, remember these are the enzymes that form the different components of our cell wall by catalyzing those covalent bonds. Those penicillin binding proteins are slightly different in gram-negative cells, and so we don't get very good competitive inhibition. Now, we can modify some of these drugs and make them more effective against gram-negative cells, but in general, they're going to be kind of our primary go-to ones if we have a gram-positive infection rather than a gram-negative. Okay, so if you need to go back and listen to that spiel, again, make sure that you do that, because we're now going to talk about some of the ways in which bacteria can get around the effects of these beta-lactam drugs. So when we start looking at bacteria that have been in the presence of these beta-lactam drugs for a long time, some of them have acquired a gene for what is called beta-lactamase. Right away, you should clue into the fact that beta-lactamase ends in ASE. Beta-lactamase is an enzyme. What do you think beta-lactamase does? It breaks down the beta-lactam ring in these drugs. So there are some bacteria that have acquired a gene for this enzyme, and they can produce the enzyme beta-lactamase within their cells. So when we have beta-lactamase produced, will our beta-lactam drugs be effective at inhibiting cell wall formation? No, it won't be. If a bacterial cell has the ability to produce beta-lactamase, in the presence of beta-lactam drugs, beta-lactamase will degrade the antibiotic. And so cells, bacteria cells, if they have this enzyme, that's a good thing for them. It allows them to survive even in the presence of these beta-lactam drugs that at one point would have been damaging to them because it would have inhibited their cell wall synthesis. But now they don't care about penicillin and these other beta-lactam drugs because they just break it down. This is one example of how we can have antibiotic resistance evolving in some species of bacteria. So in this particular instance where we are looking at antibiotic resistance, we are going to use the penicillin and beta-lactamase kind of tit-for-tat back and forth evolution as our example for antibiotic resistance. But this same idea has happened. It has occurred in a number of different species with a number of different drugs. So we'll talk about some of the ways that we used to have antibiotics that would kill or inhibit a given species of bacteria. We say that if that species was inhibited or killed by that drug, that it was susceptible to that antibiotic. But if the genetics of that species have changed and they are no longer affected by that antibiotic, that species has then become resistant to the antibiotic. One very important thing that I need to clear up, because it's not uncommon for people who come into this class to think that antibiotic resistance refers to a person's body who is, like it's your body that is resistant to the antibiotic. And it has nothing to do with your own physiology. It's really about how that drug affects that bacterial cell. If it had one point affected that cell and no longer does, we say that that cell, that species as a whole, has evolved resistance. So it has nothing to do with the human body. All right, so when we start talking about how antibiotic resistance evolves, we've got a kind of a lack in a diagram in your book. And so I've pulled this one from Google to kind of help us work through how antibiotic resistance develops. And again, we're going to use it in the context of penicillin, how we have evolved resistance to that drug in a number of different species. So here in this diagram, our purple cells are considered normal bacterial cells. They are going to be the ones who are susceptible to our drug. We're going to see that there is a single mutant in our population of normal bacteria. This mutant has acquired the gene for a resistance characteristic. In our case, this gene is going to be the gene for beta-lactamase, the enzyme that can break down the beta-lactam ring in penicillin. So this bacterial cell has no idea that it has an advantage over the other cells. And until penicillin is administered, it doesn't have an advantage. But when that drug is administered and all of these susceptible cells are killed by that bacteria, we're going to see that we have one cell that just randomly survived exposure to that antibiotic. Sometimes that happens. Antibiotics may not be 100% effective against all of our cells. But this isn't our mutant. When we look to our mutant, they have survived exposure to that as well, but it's because of that gene for beta-lactamase. This orange cell, this mutant cell with the beta-lactamase gene, broke down that beta-lactam drug. And so now we see normal physiological processes of our surviving cells taking over. They're going to divide. And before they divide, they have to copy all of the DNA within their cells. They do this and divide, just like normal. the random cell that went ahead and survived is going to copy its cellular instructions. They don't have the gene, so it's just going to be purple that we're passing on. But those that are orange reflect the beta-lactamase gene. Because that one cell had an advantage and it survived, it can now divide, passing that gene on each time it undergoes binary fission. And over multiple exposures to penicillin, we select for the trait of the beta-lactamase enzyme. And what we will eventually get over lots of different exposures to that penicillin is a whole population that is now the mutant resistant form. So this essentially is evolution. We have a susceptible population in the beginning that has evolved resistance over multiple generations, because now we have a high prevalence of this beta-lactamase gene because surviving allows us to pass it on. Bacteria can evolve resistance very, very quickly because they replicate very, very fast. We will look at more of this process of developing antibiotic resistance in detail in just a little bit. We'll talk about it more when we start talking about genetics, because that mutation, how we get that beta-lactamase gene, is incredibly important. And it's kind of a cool thing that bacteria can do to acquire some of these resistance genes. So this is just one example, but there are many different ways in which antibiotic resistance can be conferred. All of these are going to come down to changes in the genetic traits of a cell. If a cell has a gene that gives it an advantage in the presence of an antibiotic, it will live during that exposure to the antibiotic, and it will pass on that mutation to subsequent cells that it makes. Okay, so there are 4 different general ways in which antibiotic resistance can occur. The first is that we can alter the target. We can modify the target. So when we have a drug that is looking for a specific structure within the cell, well, if we change that structure, the antibiotic can no longer find its target. The example with beta-lactamase destroying beta-lactam drugs is an example of number 2. So when we destroy the antibiotic before it has a given effect on whatever it is that it's trying to do, well, then you've degraded the antibiotic, and it doesn't matter that it's there if the cell can get rid of it. A bacterial cell sometimes has the ability to add on functional groups to the antibiotic compound, and so it can no longer target whatever process it is trying to disrupt within the cell. My favorite one is number 4. So a bacterial cell can acquire a gene for a pump that can be embedded within its microbial membrane. Well, if you have a gene for a pump that can pump out the antibiotic, as soon as that antibiotic comes into the cell, well, you kick it right back out. And so there's no antibiotic that's actually in the cell. So bacteria have all kinds of ways to try and survive exposure to antibiotic resistance. And when they have a change in their genetics that allows them to survive, that gives them an advantage. So I'd like to watch this experiment that was conducted at Harvard. It shows the evolution of antibiotic resistance in a particular bacteria. Let's take a look at this.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands we put a normal petri dish thick agar with different amounts of antibiotic. On the outside there's no antibiotic. Just in from that there's barely more than the E. coli can survive. Inside of that there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So antibiotic resistance will arise when we overuse antibiotics. So when we start talking about how those genetic changes are going to take place, if there's no selective pressure for our cells, we won't see this spreading of these resistance genes. But nowadays we are seeing a great number of microbes becoming resistant to the antibiotics that we use because they are evolving. They are evolving ways in which to survive. This is what they're going to do. So we have to be careful that we don't overuse antibiotics. The truth is that penicillin, which was identified and really started becoming widely used, was in the 1940s. And there are so many species that are resistant to penicillin right now, not just through the evolution of beta-lactamase, but a number of those other mechanisms that we kind of broadly talked about. So within a very short period of time, we have made this particular antibiotic virtually useless, and we're doing the same thing with a number of other antibiotics. So make sure that we kind of as a society are going to be using our antibiotics appropriately, only when they are needed, and we're taking the full course of antibiotics for when a doctor prescribes those to you. Make sure that you're finishing them up. Even if you feel better a couple days before the end of your dosage course, make sure you take all of them. Okay, so that's my PSA for this particular video. Okay, so penicillins, there are a whole bunch of different types. We saw where that side chain was that we can change and alter. We have 5 different general groups based on these side chains. Cephalosporins are considered to be semi-synthetic. They are naturally produced, and then we can alter them artificially in a lab. We have multiple generations of these as well. So it's kind of, again, this tit-for-tat evolution. Some of our beta-lactamase enzymes are not going to degrade certain types of cephalosporin. So we say that our drug is now resistant to the enzymes that confer resistance to our microbes. And I know that that's kind of confusing, but as our bacteria evolve, we have to constantly change our antibiotics so that they're still effective. And the truth is that we have a limited number of changes that we can make before they simply won't be effective. Okay, so we're now kind of moving into the matching part of this cell wall targeting drugs. So vancomycin, again, we've talked about before. We are going to see vancomycin inhibiting the cell wall synthesis. Recall that the vancomycin drug is going to bind to that string of amino acids, like on that last amino acid, it'll stick there. And so what it does is it alters essentially the substrate that our PBPs are looking for. So when we look at vancomycin up here, it says that it binds to the amino acid side. So those amino acids are really where we're trying to catalyze that covalent bond. If we can't do that, the cell will burst as well. Bacitracin works inside the cell, and it prevents the NAG and NAM disaccharide from actually getting out of the cell to where our cell wall is going to be assembled. So, you know, if you think of those individual NAG and NAM as like bricks, if you don't have the bricks, you can't build a wall. So bacitracin keeps those components inside the cell, and they're not available to build that wall. All right, before we jump into our next groups of antibiotics, I would like to review the central dogma that we've talked about before. How do we use DNA? Because how we replicate and use DNA is going to be important targets, because these processes are things that bacterial cells have to do. But we've got to be really tricky with this, because these are processes that your cells do as well. So we've talked about DNA. We said that we use it two ways. Before we undergo binary fission, we have to copy the DNA. Do you remember the name of the enzyme that is responsible for reading the chromosome and making a copy of it? The enzyme that reads DNA and makes new DNA is called DNA polymerase. So DNA polymerase has to act to copy that chromosome, and then we take those two copies and put them into two different daughter cells in binary fission. We do that time and time and time again. DNA polymerase is incredibly important, but you know what? We have DNA polymerases too. When we use DNA, we're going to take that DNA and we're going to make an intermediate molecule, RNA. Do you remember what the name of the enzyme is that does that? The enzyme that reads DNA and makes RNA is called RNA polymerase. Again, make sure that you use the full name of both of those enzymes, because if you just put polymerase down, I don't know which one you're talking about. But you know what? We have RNA polymerases too. What's the structure that reads the RNA 3 nucleotides at a time and strings the correct sequence of amino acids together? Ribosomes. So we have ribosomes. Bacterial cells also have ribosomes. Structurally, these guys are different. I'm not interested in you knowing the different numbers that reflect the different subunits of the ribosomes, but the two subunits of human ribosomes are different than the two subunits of prokaryotic bacterial cell ribosomes. Okay? So that's kind of like where we're at so far, to catch you up and kind of refresh that important process. So now we're going to look at drugs that target copying DNA. So if you can mess up the synthesis of DNA, well, when you have that parent cell and you're trying to divide it into two daughter cells, those daughter cells don't have the correct genetic information, which if you're a person who has an infection is a good thing. If you're a bacterial cell, though, that's a bad thing. You need your instructions. So if we can screw it up, we can help to clear out that pathogen. So one of our groups of drugs that does this, that targets DNA synthesis, is called metronidazole. So it is going to bind to the DNA, and it's going to interfere with the synthesis, and it's ultimately going to lead to breaks in the bonds that hold the DNA together. Well, structurally, our DNA is the same as prokaryotic DNA. So, gosh, how do we achieve selective toxicity? Metronidazole is a drug that has to be reduced inside a cell in order to be effective, in order to actually bind to the DNA and do the damage. So this drug can only be activated in anaerobic organisms. This means that our cells, which are strictly aerobic, won't activate the drug in our own cells. So this is how we achieve selective toxicity. But the fact that it has to be activated in an anaerobic organism also limits the number of species that it can be utilized against. So we can only utilize this guy if we have an anaerobic species that will actually activate the drug. Sulfonamides is another one of those bold terms at the end of the chapter, so this one we're going to spend a little bit more time with. Sulfonamides are also sometimes called sulfa drugs. So this group of drugs is structurally similar to a component of folic acid. So folic acid is a substance that is required for the synthesis of long nucleic acids. So we have to have folic acids to make DNA, to make RNA. Well, humans don't make our own folic acid inside our body. In order to obtain the folic acid that we need to make our nucleic acids, we have to ingest it. So you may be familiar with folic acid if you take a vitamin, especially women's, like vitamins that are specifically for women. They will always have 100% of the folic acid in that vitamin. And it's because we want to ensure women who are of childbearing age, should they become pregnant, we want to make sure that they have an ample supply of folic acid, because they're going to have a human being developing within their body that is going to require that folic acid to synthesize a whole bunch of new DNA as that embryo develops and grows. So this is likely where you have had folic acid that you may be aware of. It's something that it's important to consume for humans. Bacteria, though, don't obtain fully formed folic acid the way that we do. They assemble it. This is where the selective toxicity between human cells and bacterial cells come in. So normally what happens when our bacteria are going to make their folic acid, there's an enzyme that's going to do it. And it requires 3 different substrates to come in. One is called pteridine. The other is called PABA, which stands for para-amino-benzoic acid, shortened to PABA. And then the other component of folic acid is glutamic acid. So these 3 components come in, and our enzyme catalyzes the formation of folic acid. So now the bacterial cell has its folic acid. It makes the folic acid. Well, if you can use a drug that prevents the formation of folic acid, you can essentially stop the synthesis of DNA and RNA. So what happens is that this para-amino-benzoic acid, PABA, is going to have a similar structure to our drug, our sulfa drug that we're going to administer. So what happens is that we have pteridine and glutamic acid come in, but this sulfa drug is sufficiently similar to PABA that it will sit in that active site. And that means that our substrates can't get in quite the right formation, and the enzyme cannot catalyze the formation of the folic acid. So in this instance, our sulfa drug is going to block the formation of folic acid. We're not going to have the bacterial cells having the folic acid that they need in order to correctly synthesize their genetic instructions. If they don't have their genetic instructions, they don't have the information to make the stuff that they need. All right, let's go back to the ones that we're just going to touch on briefly. So we have a limited number of drugs that will target RNA synthesis. One of those is called rifampin. This particular drug binds to strictly RNA polymerase, so it won't bind to our RNA polymerase. And what it's going to do is it's going to prevent the reading of DNA and making that RNA in a process called transcription. So transcription is the name for reading DNA and making RNA. We'll talk about that more in a bit. But if we shut that process down, well, you don't get your proteins and you mess up all of the different processes that are going to happen within the cell. So this is a drug that is sometimes used to treat tuberculosis infections and certain types of meningitis. The image that is pictured to the right, your book has chosen, and it kind of makes me giggle. This particular drug has a completely harmless side effect of changing a person's urine to a bright orange color. So that's what they're showing you there, is that this particular drug will, it's harmless, but if you have a patient who's going to take rifampin, you should probably give them a heads up so that the next time that they pee, they don't freak out, because it's going to be bright orange. All right, so if we can copy our DNA and we can use it to make RNA, what if we inhibit the reading of that RNA to make our protein? So there's a number of different drugs that can inhibit protein, but we're going to look at two broad groups that each target different subunits on the bacterial ribosome. Remember that those ribosome subunits are different in bacteria than they are in humans. This is how we achieve selective toxicity. So aminoglycosides, which include things that you might know, streptomycin, gentamicin, these are considered broad spectrum antibiotics. So they are going to inhibit protein synthesis in a whole bunch of different microbes. These are bactericidal, meaning that they will actually go in and kill the cells. These drugs bind to the smaller of the two ribosomal subunits, the 30S subunits. And the reading of RNA has to occur 3 nucleotides at a time. Well, this particular class of drug causes the misreading of that RNA. So we're no longer reading it at the correct 3 RNA nucleotides at a time. Because this one has some pretty severe side effects, and they're not uncommon. Usually this is not like our first go-to set of drugs, especially because it's a broad spectrum bactericidal. Even if you don't have a patient who suffers these more severe side effects, this class of drugs oftentimes will really disrupt normal flora and can lead to some really awful opportunistic infections. But some of the more long-term damaging effects of use of this class of drugs is that it can affect hearing. So this drug can damage the auditory nerve that feeds information about sounds to the brain. Additionally, this particular drug is pretty hard on the kidneys. Macrolides are another group of antibiotics that include erythromycin. This is, again, a broad spectrum drug, but these are bacteriostatic. So they are going to inhibit bacterial growth sufficiently so that our immune system can kind of get a hold on things, but they're not necessarily going to kill the cells outright. So we have this one that's going to bind to the larger of the 2 ribosomal subunits. Here we're going to cause misreading, not misreading, we're going to prevent the correct amino acid from coming in. Even though we're reading the set of 3 nucleotides at a time correctly, we're going to have the amino acids that should be brought in and assembled into that nice long protein just not being brought in the way that they should be. If you are a person who is allergic to penicillin, oftentimes this is kind of a first backup to that class of penicillin drugs. It's not uncommon to have allergies to a lot of these antibiotics simply because they are compounds that are found in nature. So penicillin is a really common drug allergy, but sulfa drug allergies, as well as a number of these other ones, are important things that should be shared with your doctor so that they can consider that in addition to all of the other stuff that they're considering as they prescribe certain drugs to treat infections. All right, with that, we wrap things up. So make sure that you get your worksheet done for Chapter 13. The reading is listed there. Some of this information you won't be reading. You'll be relying on the videos for. We've got lots of review questions to test your knowledge. And then remember to spend some extra time understanding how penicillin, cephalosporins, and sulfonamides work. The other non-bold words will be matching on your exam. Let me know if you have any questions.